Hey guys, today we're diving into something called base editing and how Verve Therapeutics team is working to tweak one gene called PCSK9 to help keep our hearts healthy. By the end, you'll see how a single treatment can change millions of lives. Let's jump in. First up, let's understand how genetic instructions become proteins. Genes are stretches of DNA that carry instructions. Those instructions get copied into mRNA, which a molecular factory called a ribosome reads to build proteins, the tiny machines that do most jobs in your body. Now that we know how proteins are made, we need to break DNA down even further. DNA is made of 4 bases, A, T, C, and G, like letters in our genetic alphabet. A always pairs with T, and C always pairs with G. Changing just one letter can change what a gene does. Cells talk to each other by sending signal molecules that bind to receptors using special antennae on the cell surface. Once a signal docks, it'll tell the cell to grow, divide, or take in molecules. The idea of receptors will become important when we meet our star protein, PCSK9. Here's PCSK9. It's a protein that grabs LDL receptors, the cell's cholesterol doors, and drags them away. With fewer doors, more bad LDL cholesterol stays in your bloodstream. Now you see why PCSK9 is a prime editing target. Too much LDL sticks to artery walls, building a plaque that narrows vessels. Over time, this raises your risk of heart attacks and strokes. So how do we stop PCSK9 from taking away those receptors? Let's turn to gene editing. CRISPR-Cas9 is like a molecular scissor guided by a GPS map, which is a guide RNA. It finds the right DNA spot and cuts the strands so scientists can remove or insert code. But it still makes double strands break. So there's an even better tool next. Base editing acts like a pencil that swaps one DNA letter for another right inside the PCSK9 gene. That single letter change introduces a harmless typo so that the cell makes a non-functional PCSK9 protein. Without working PCSK9, the liver cell's LDL receptor doors stay on the surface, letting it clear more bad cholesterol from your blood. To edit genes inside a living body, called in vivo, scientists package the base editor machinery into tiny fat bubbles, or lipid-nanoparticles, that home in on liver cells after injection. Speaking of which, here's how that looked in people. In the HER2 trial, volunteers got a single infusion of VERVE-102 at a low, medium, or high dose. Researchers then tracked safety and cholesterol changes over the next month. Ready to see the results? Here it is. By day 28, high-dose participants saw an average 59% drop in the LDL cholesterol, with no significant side effects reported. That's a huge win for a one-and-done gene therapy. Now, why does that matter for the real world? Imagine one shot cutting your lifetime heart disease risk over by half. No daily pills or regular injections needed. That's the promise of PCSK9 base editing. And the story doesn't stop here. After those fantastic Phase 1 results, Verve Therapeutics is gearing up to dose its first patient in Phase 2 later this year, taking us one step closer to a real treatment for millions of people. Let's review over our journey. We saw how bases become proteins, why DNA bases matter, how receptors work, why PCSK9 is key, how base editing precisely swaps letters, and that the HER2 trial delivered a 59% LDL drop safely in just 1 month. Thanks a lot for joining us on today's video. We hope you had a lot of fun and learned something new. If you like this video, don't forget to like and subscribe for more Biology Bytes. If you care about helping other kids learn, check out Them USA. They bring reading and learning to children across India and help build a better world. Visit ProthamUSA.org to see how you can help. See y'all next time. Bye!